Hey guys, how's it going? Can you guys hear me?
Can hear you now.
Okay. I unfortunately left my normal headphones at my girlfriend's mother's house, so I am tethered to my computer this morning.
Mhm.
So if you guys look at Moodle, you can see that I've posted the food blogging instructions. So I've actually given you 4 specific prompts to respond to. They're all centered on fall break, which, as you realize, we're not getting this year. And so typically I would have given some prompts having to do with kind of how your diet has changed while you're at William Jewell College, eating in the cafeteria and things like this, and then how those food habits change when you go home, because they inevitably do. But given that you're not going home for fall break this time, I've kind of altered the prompts to reflect that. You've probably been eating in the caf for a while now, and I'm guessing that that might get old after a while, or it might not, depending on kind of what your tendencies are in terms of using the cafeteria. So anyway, they're all focused on your experiences in the cafeteria, and so it tells you when to respond to each prompt. One of them is first response during the first week, second response during the first week, first response during the second week, etc. So that's up there. We have an exam on Friday scheduled, and so I'm working on putting together a review sheet for you. Not sure when I'll get that done, but I'll post it as soon as I have it done. Some people also mentioned wanting to have a review either in person or online on Zoom, and I would be open to doing that. What I don't generally like doing that the night before, because it doesn't really give you much time to then take whatever you learn from that experience and incorporate it into your prep. I have something in my eye. So if we could do that on Wednesday, that would probably be more useful to you than doing it on Thursday, simply because it'll give you adequate time to take whatever you learn and put it to good use. I am out of class at 1:35 on Wednesday, so I could do it anytime after that. So I'm open to whatever.
If it is on Zoom, will you record it just in case people can't make it?
Sure, but let me just tell you this, that last year when I was doing this class, I was already recording lectures. So if you go online, you can see lectures from previous years online. And when I ran a review session for the first exam last year, a student asked if it was going to be recorded, and I said yes. Sure, I can do that. And then only two people proceeded to come to the review session. So the review session wasn't terribly useful because only 2 people attended. And so, yeah, I can do that. But if in the process of doing that, you all just say, oh, well, Dr. Klawinski's recording it, I'll just look at the video later, and nobody shows up for it, it's going to be a really uninteresting video. And once again, the only reason I'm saying this is because I don't think that you guys will behave this way. It's just that that's how last year behaved. So a review session is best when a bunch of people come because a bunch of people have questions and everybody has a different question. So you actually get a lot done rather than one or two people coming. And I'll do it for one or two people. It's just, like I said, less useful. So yes, Marcus, I will do it. But hopefully it will be useful. Do you want it in person? You want it on Zoom? When do you want it? Do you want it? Or do you just want a sheet of topics?
If we do the review, will we not get a sheet of topics on Moodle?
No, I can do everything because that's what we do here.
Okay.
Yeah, I can do that. It's easy enough for me to sit down and think through what we've covered so far. Actually, it's kind of useful for me to do that because it forces me to revisit the material as I get ready to sit down and write a test.
I think a review on Wednesday would be helpful. Personally, I don't get done with my Wednesday stuff until 4. So if it's before that, I wouldn't be able to make it, but I think it would definitely be beneficial.
Okay, well, why don't you guys email and back and forth amongst yourselves and arrive at a date date and time that works for you, and then just let me know. I will actually send you. Let me see. After class today, I'll send you a copy of my schedule, the schedule that sits on my door outside my office door, so that you'll know exactly when I'm free as well, and then you guys will kind of know what times are available. But I'm reasonably flexible. I normally have a department meeting on Wednesdays at 3, but we met last week, so I can't imagine that we're going to be meeting this week. So I should be free as soon as I get out of evolution and ecology class at 1:35. All right. How are your preparations going for your group presentations on the genetic engineering report?
Actually, I wanted to propose something. So I'm feeling kind of overwhelmed with everything, with the test and then the presentation being that next following Monday.
Yep, I could understand why that would be the case.
Yeah, so considering I want to focus well for the test and focus everything I need to do this week on that, it just seems like a lot to pack in so we have a good presentation that Monday. And so I was wanting to feel out everyone else and how they were feeling about the time.
Oh, come on, Olivia. Don't you know that college is all about stress?
Yeah, I know.
I'd agree with that, especially since I work a lot on weekends. I wouldn't be like if our group was trying to do a lot of our weekend from between the test and the presentation, I wouldn't be able to contribute as much.
Okay. Well, I had already kind of broached this topic, which is why I think I said last week, if things are not going as quickly as you hope they would or whatever, the nice thing about the presentations is they're relatively fluid. I don't want to push them off until the very, very end, but I also we don't have to do it on Monday if we don't feel like it. So in this way, we do have some flexibility. So what do you guys think would be a reasonable date? I'm looking at the schedule. So I would like to probably get it done before October is done, simply because that'll give you some time to digest before you actually have to write your paper.
So maybe next Wednesday or Friday.
So next Wednesday or Friday, that would be the 23rd of October. Would you guys think you would be ready by then?
I think having it that next Friday would give us it gives us a good week to really focus on our presentations and polish them.
Okay, then what we'll do is we'll move food webs up to Monday. We'll move the historical roots of modern agriculture up to Wednesday, and then we'll have well, let's see, let's do this. Rather than splitting material, so we have a lot of, so you guys are going to be so happy about this. We're about to leave science. And we're going to move into history. So we have like 4 days actually of looking at history. So why don't, rather than starting on history and then breaking that up with some presentations and then going back to history, why don't we just put off the food presentations until the 30th of October? That'll give us an opportunity to talk about food webs, talk about the history of agriculture, and then in a break between topics, we can talk about GE foods. Does that make sense? Okay, so then let's set that. I'll revise this and post a new schedule, but write down on your calendars for right now, Friday, October 30th will be the new date for genetically engineered food presentations. The other thing that does is it gives you the opportunity to have enough time to get something together and put together. And if you want to run through it with me, you can. If you want to run through it with maybe other members of other groups in the class, you can. You have an adequate amount of time to perform well on that particular assignment. So Jackson has practice until 6:30. Would you guys want to get together in the evening on Wednesday?
Yeah, that works for me. I know in the chat, a lot of people said they were free kind of after 2 or 4 and Jackson 6:30. So I think that'd work.
Okay. I normally ride on Wednesdays. So would like 8 in the evening work? I'll be done with my ride by about 7:30. You may see me in my cycling gear, but you know, that's the way it goes. Your fault for scheduling something in the evening.
I think it works great.
What's that, Olivia?
I said I think it works great for everyone, with it being in the evening.
Okay, 8 o'clock. You want it in person or Zoom?
Zoom would be best for me, so I don't have to drive to campus.
Well, I can Zoom it to any. I mean, I can log on to Zoom even if you're not, even if I'm doing it in person with other people. What would your preference be? All right, Zoom it is. If you have a different preference, you know, let me know. Drop me an email or whatever. Yeah, so I saw the chat lighting up, so I thought I would go see what you guys were talking about. And you're talking about times. So 8 o'clock Wednesday on Zoom. I'll send you a Zoom invitation unless somebody says, oh, I'd much rather do it in person, then we'll do that. On that front, so since I wasn't planning on talking about this, it seems that Missouri is leading the nation in the increase in new cases. I saw a map over the weekend and Missouri is lit up bright red when you look at the percent change in new cases week by week. So for the previous 7 days, we have among the highest percentage increase in rates of COVID-19-ness. I was also listening to some Kansas City, Missouri public health people on the radio last week, and they were being asked to explain why the number of cases has been declining for a number of weeks in Missouri, but the number of hospitalizations and the number of deaths has been increasing. And their explanation was that there's actually been a slowdown in testing. So while the number of cases has gone down, that's because people as a whole are doing less testing, because as the testing has gone down, of the people who have been tested, the positivity rate has creeped up to like 16%, which is really high. So 16% of all people who have been tested, tested positive. And if you had a broader testing protocol, that positivity rate would probably be lower. And so the reason for the decline in cases and yet the increase in hospitalizations and deaths is actually due to a slowdown in testing. So continue practicing COVID protocols, and yeah, it's not getting any better seemingly here in Missouri, in our neck of the woods. So please continue to be careful around your friends and family and things like this. Somebody that I bicycle with, who I haven't actually seen in a couple of weeks, texted over the weekend that her husband tested positive for COVID and she's not going to be seeing any of us for a while because she's also showing symptoms, blah, 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 blah. So I myself am trying to be careful to the degree that I can. So be safe. All right. Any questions over what we were doing last week? Just so you know, nutrient cycling is one of the things that our biomajors finds the most difficult to wrap their heads around. So this stuff that we're going through today is actually reasonably complicated and takes some time to wrap one's head around. So I want to make sure that we're taking an adequate amount of time and we're not rushing through it. And so as you have questions, as I'm going through it, by all means, stop me and ask me to back up on stuff. So can you guys see my screen? Okay. So we had been talking about the carbon cycle, and we had just started talking about the nitrogen cycle. So nitrogen is hugely abundant in the atmosphere. But the problem with that form of nitrogen in the atmosphere is that it's a triple bonded molecule. And with each additional bond that you get between two atoms, the bond strength gets stronger and stronger. And so as a result of this, it's very difficult for organisms to get access to nitrogen because so much of the nitrogen that we have is just simply inaccessible to most organisms. The only organisms that are capable of breaking these bonds in nitrogen are bacteria, generally different types of bacteria. And the reason that they can do it is that they have specific enzymes that enable them to break this triple bond of nitrogen. And so when we look at nitrogen fixation, we're basically talking about taking nitrogen in the atmosphere, gaseous form of nitrogen N2, and we're going to break those bonds and combine nitrogen with hydrogen to create ammonia. And this is the first step in getting nitrogen into ecosystems and accessible to organisms. So this is generally carried out by bacteria. We talked about last time. Rhizobium is a really important one. Many of these bacteria are found in association with the roots of plants. And so I talked about legumes being high in protein content. One of the reasons that they're so high in protein content is that the legumes have these rhizobium bacteria that are found in the root nodules of the plants. Hold on just a minute. So if you go in your textbook to the chapter on legumes, which I believe is chapter 13. Yep, chapter 13. When you go there, you're going to see in figure 13.2, there is a photograph of a root of a legume plant, and you can see all of these little balls that are structured all around the root. Well, when you cut those open, they appear a pinkish or reddish color, and the pinkish or reddish color is there because they have an oxygen absorbing molecule called leg hemoglobin. And the reason that they have leg hemoglobin, let me write leg hemoglobin. I'm going to put it, actually, I'll just put it in the comments since I'm tethered here. Except that I can't access the comments when I'm in presentation mode. Darn, okay. Leg hemoglobin. I'm going to write it on the board. Can you guys see that?
Not really.
My camera seems really weird. Why is everything so washed out? It doesn't help that the sun is shining right through my window. Is that any better? Can you see that better? No? Okay,
Yeah.
all right. So leg hemoglobin. Leg hemoglobin is basically a type of hemoglobin. So just like you have hemoglobin in your bloodstream, these plants have hemoglobin in these root nodules. And hemoglobin is basically a molecule that is designed to carry oxygen. And so it has an iron atom at the center of it, and it binds with oxygen and carries oxygen around. Well, these soil bacteria that end up being found associated with the roots of plants, they do really well in low oxygen environments. And so one of the ways in which the plant facilitates these organisms living in association with them is they have a molecule that takes up oxygen from the root nodules that then creates an environment that these bacteria need in order to live. And so there are bacteria found in association with plants, and there are also free-living soil bacteria that are capable of converting nitrogen as well. Many of these are what we call heterotrophic. Heterotrophic just means that they use lots of things as an energy source, but some of these are autotrophic, meaning that they use sunlight as an energy source. But this nitrogen ultimately gets made available to plants for the production of amino acids because these bacteria are doing this first step of conversion of nitrogen from gaseous nitrogen N2 to ammonia NH3, a nitrogen with three hydrogen molecules attached to it. And so in this Bohr model of the atom that we just saw, you see that there are 3 bonds that are covalent bonds between these 2 nitrogens. When you split nitrogen apart, there's going to be a hydrogen that attaches to each of these three unpaired electrons. So the enzyme that they have that enables them to do this is called nitrogenase. Remember that all enzymes end in ASE. So nitrogenase is the enzyme, and it contains molybdenum and iron, and it uses ATP. The reason I mention molybdenum and iron is that there are oftentimes trace elements that you have to add to soil in order for organisms, in order for plants to grow well. And many of these things, they don't have to be there in great abundance, but they do have to be there. And iron, for example, is one of these. And so you'll sometimes see people amending soil on farms to include what we call trace elements that really don't have to be present in great amount, but have to be present. And in some cases, it's because of things like nitrogenase. So it requires a lot of energy. It takes about 10 grams of glucose to fix 1 gram of nitrogen. And that's a hugely energetic input to get nitrogen fixed. And this is actually one of the problems with synthetic fertilizer. We can create synthetic fertilizers through industrial fixation, which is what Fritz Haber did in inventing the Haber-Bosch process. But one of the problems with doing that is that it's an amazingly energy intensive way of going about making nitrogen-based fertilizers. And so when you look at the economics of a farm, for example, you know, they're buying tractors and they're buying seed and stuff like this, but an amazingly high proportion of the cost of running a farm is actually buying synthetic fertilizers, because they tend to be expensive, because they tend to be very fossil fuel intensive, because it takes a lot of energy, especially for the conversion of nitrogen from gaseous nitrogen to ammonia. Which is also why the carbon footprint of food is pretty high. So what happens with nitrogen fixation? Oh, here's a picture of some root nodules. What happens is that the root hair grows out, rhizobium, which is a soil bacterium, becomes associated with the root hair, and then the root hair slowly grows around the bacteria, and the bacteria then become associated with these nodules. And so even in the case of rhizobium, which has these associations with plant roots, forgive me, they start out in the soil and ultimately end up inside the plant root, but they begin there as free living soil bacteria. And so the plant actually produces plant material around the bacteria that ultimately ends up developing into these nodules. And so when we think about two organisms living in really close association like this, we refer to that as a symbiosis. So a symbiosis is just the word that we use for describing when two organisms live in close association with one another. Now, oftentimes, the only symbiosis that you learn about in public school is mutualism. But mutualism is just one type of symbiosis. And so mutualism is a symbiotic relationship where you have two organisms living in close association with one another. And in this case, both organisms benefit from it. So I'm going to write some things up on the board and then I'll come back to you. Okay, can you guys see that all right? Okay, so what I've written on the board is a series of interspecific interactions. Mutualism, you probably learned about at some point in your public school education. Mutualism is a symbiotic relationship where both organisms benefit. And in the case of these guys, it's clear what the plant is getting out of the deal. What is the plant getting out of the deal? What benefit is the plant getting from this association with these bacteria?
Aren't they getting the nitrogen?
Yeah, they're getting the nitrogen. So the bacteria are fixing nitrogen. That nitrogen is being absorbed by the plant roots. So what the next question then is, what is the bacteria getting from the plant if this is a mutualism? What are plants in the business of doing? It's one of the things that makes plants great and distinguishes them from animals.
Creating oxygen.
What's that, Blair?
Creating oxygen.
It's they create oxygen in the as a byproduct of doing this other thing that I'm thinking of.
Photosynthesis.
They do photosynthesis. So one of the things that they do is they create sugar, and they generally create more sugar than they can need, than they actually need, which is why they grow. And they store a lot of that sugar in their plant tissues in the form of cellulose. But because they oftentimes create more glucose than they actually need, and they end up storing a lot of it in their plant bodies, they have glucose to spare in many cases. And of course, what do you need a lot of in order to fix nitrogen?
You need a lot of glucose.
You need a lot of glucose. And so what happens is these plants, they receive nitrogen from the bacteria, and what they give up as a result of receiving that nitrogen is they give up some glucose as a result of this. Those glucose feed the bacteria, and the bacteria need that energy because the bacteria need a lot of energy in order to facilitate this transformation from gaseous nitrogen into ammonia. And so when we think about what partners are getting what in this particular mutualism, plants are getting nitrogen, bacteria are getting glucose. So back to these other types of symbiotic relationships, all of these could be considered to be symbioses, but only one of them is mutualism, when both organisms benefit from the interaction. Under parasitism, a parasite benefits, but the host is negatively affected. And so you can think about all of these interactions in terms of what are the consequences for the two species that are involved. So if you get a parasite, it's going to draw energy and nutrients away from you. And so the host is negatively affected while the parasite is positively affected. Competition is a situation where both species are negatively affected. Imagine two shoe stores competing for the same customer base. The fact that one shoe store is in the presence of another shoe store makes basically they have to divide the market that they have for selling their shoes between the two of them. And so both of them are worse off than they would if they didn't have competition. Predation is another plus minus situation. In this case, rather than the parasite and the host, the one that benefits from predation is the predator, and the one that is negatively affected to the point of death, generally speaking, is the prey. And then we have this thing called commensalism, where one species benefits from being in association with another species, but there's really no measurable effect on the second species. And remoras that attach to sharks is one example of this. I work with a species of gecko that tends to live around human habitations, and they benefit from those human habitations, but they really don't have any effect on humans. So that's another type of commensalism. So interspecific interactions, most of these would be considered to be symbioses, but not all symbioses are mutualism. So it's one of those, what do they call those things when all mutualisms are symbiotic, but not all symbioses are mutualistic, is one of the things I want you to grasp from that. So we've already talked about the benefit that plants get from this relationship with nitrogen fixing bacteria. They get access to ammonia, and that enables them to build proteins. So where nitrogen is concerned, humans have increased the global nitrogen fixation by more than 3 times. And they've done this by 3 different mechanisms. 2 of them are purposeful, and 1 is just a byproduct of just us being humans. So one of the things that we've done that has increased global fixation is by cultivating nitrogen fixing crops. So when we grow soybeans or peas or lentils, or even when we grow cover crops that are nitrogen fixing cover crops on farms. So right now, farmers are out there harvesting. They've been harvesting stuff in this area for about the last month and a half now. What a lot of these farmers will do is they will then go back over the winter and they'll seed their fields with something like clover or vetch or something like that. Clover and vetch are both nitrogen fixing plants. They'll let those plants grow over winter, and then in the early spring, they will plow those plants under. They're not growing those plants for a crop. Those plants are just a cover crop. What they do is they cover the ground fairly densely, which then prevents the growth of other kinds of weeds. And because they're nitrogen fixing, when they plow them under in the spring, all of the nitrogen that is in the bodies of those plants will then become incorporated into soil, which will make next year's crop grow better. So whether you're cultivating nitrogen fixing crops to actually harvest and eat, or whether you're using them as a ground cover and a soil amendment, doesn't really matter. We plant tons of nitrogen fixing crops. Another way in which we have increased the fixation is through the Haber-Bosch process, the creation of synthetic fertilizers. But another way in which we have increased fixation is via fossil fuel emissions. And so in the fossil fuels that we use, nitrogen, oil, natural gas, coal, these are the old dead bodies of plants, essentially. And so in all of those fossil fuels contain nitrogen. And so there's a certain amount of the emission of nitrogen in the burning of fossil fuels that puts nitrogen in the atmosphere. And this nitrogen that is in the atmosphere isn't N2, it's different types of nitrous oxides. So NO3, NO2, these types of compounds are floating around. And once again, those compounds are absorbable by plants. And so, so we end up getting a certain amount of atmospheric deposition. It comes back down to the soil in the form of rain, snow, fog droplets, and things like this. And also we get quite a lot of atmospheric deposition of these particles just falling down to the earth as dust. And in some cases, this is significant and measurable. And so I have a colleague in Puerto Rico who studies dust from Africa. And what happens in Africa is they get these big sandstorms, these big dust storms at certain times of the year. And the winds will actually blow that dust all the way across the Atlantic Ocean. And in Puerto Rico, where I've worked in the past, that dust falls out of the sky, both in rainfall, but also just in dry fall dust falling to the ground. And it makes up a significant nutrient input into the forests of Puerto Rico, this translocation of dust from Africa to the New World. I think it was earlier this summer, actually, that there was a big dust cloud that blew over from Africa and actually influenced the color of the sunsets and stuff here in Missouri, which was a very unusually large dust storm. So once it gets into living organic matter, many of the kinds of transformations that nitrogen goes through are similar to the ones that carbon went through. Live organisms ultimately die, and that dead organic matter becomes part of the soil. And so you have nitrogen moving from living organisms to the soil as those organisms go through the process of decomposition. But the decomposition process is different chemically for nitrogen than it is for carbon, because you're dealing with nitrogen containing compounds. So this dead organic matter gets dissolved in the soil into what we call dissolved organic nitrogen. And this dissolved organic nitrogen can then leave the system through leaching. So I'm gonna have to go back to the board now. I can't hear you because I don't have my headphones on. Let me see if, is this going to provide too much feedback? Can you guys hear me? Somebody talk to me.
I can hear you.
Okay, are you getting feedback? Okay, cool. All right, well, good, that works. I'm a little freer. So when we talk about leaching, what we mean when we talk about leaching is, remember that water, which is two hydrogens bonded to an oxygen. Remember that oxygen is an electromagnetic atom. So here's where all that stuff about electromagnetivity and stuff like this becomes important. Remember that oxygen, because it's highly electromagnetic, it tends to pull electron density towards itself, which makes it partially negative, and it makes each of these hydrogens partially positive. Well, nitrogen is also one of these electronegative atoms. And so when nitrogen is bonded to hydrogens, or when nitrogen is bonded to oxygen for that matter, what can happen is that these also become polar molecules as well, where nitrogen is a partially negative. Well, actually, it's a better illustration with ammonia. Nitrogen is partially negative, and these hydrogens, once again, are partially positive. And because these hydrogens are partially positive, hydrogen bonds can get set up between the oxygen of water and the hydrogen of ammonia. And when this happens, as water percolates down through soil, what will happen is that water will latch onto these partially positive hydrogens that are then attached to a partially negative nitrogen. And it will literally, as it percolates through the soil, it'll just latch onto those nitrogen containing compounds and suck them out of the soil. They'll get attached to water, and as water flows through the soil, nitrogen flows through the soil, and then it flows out of the soil into a stream, and then flows downstream into a river. That river flows into the ocean, and those nutrients are gone from that ecosystem. That process is called leaching. So So leaching is just the movement of nutrients through soil, because those nutrients are polar molecules, meaning that they have a difference in charge on them. And those polar molecules are pulled out of the soil due to the movement of water through the soil, because water itself is also a polar molecule. So whenever you see the term leaching, this is what is meant by it. And one of the problems with leaching for nitrogen is that nitrogen is limiting in soils. And one of the reasons that it's limiting in soils is because it tends to leach out of soils easily. Once this organic nitrogen gets into the soil, it can be leached out through that process, but it also becomes food for the major decomposers. And these major decomposers are bacteria for animals and fungi for plants. And so this decomposition takes whatever form the dissolved organic nitrogen is and basically converts it back into, in many cases, gaseous nitrogen. So when we look at this, we take dissolved organic nitrogen, and through the process of ammonification, it gets converted into ammonia and then into ammonium. And then that ammonium becomes basically just a part of the soil. And so microbes do this, bacteria and fungi do this. And so they take in the organic nitrogen and they secrete ammonia, which is then converted into ammonium. And this is all determined by a variety of different things. Generally, organisms like bacteria, they grow fastest in warm temperatures. They grow fastest in moist environments. They also have trouble sometimes getting access to the nitrogen that is inside of a material. So for example, this carbon to nitrogen ratio, if there's a lot of carbon in the tissues of an organism, the bacteria have to break down all that carbon in order to get access to the nitrogen. And this is actually one of the challenges of decomposing plants. Plants have a lot of carbon in the form of cellulose, and that carbon basically protects the nitrogen that is inside the cells of the plant, such that the first thing a bacterium has to do is it has to break down the cellulose in order to get access to the cells. And then after it has access to the cells, it can then draw that nitrogen out. And so the carbon nitrogen ratio of the things being decomposed becomes important. The more carbon there is, the harder it is to get to that nitrogen. So if you look at animals like us, we have relatively low carbon to nitrogen ratios. We have a lot of nitrogen relative to our carbon than other organisms do. Now, it's still skewed towards the carbon side. There's a lot of carbon in us, but relative to that carbon, there's a lot of nitrogen in us. A plant, on the other hand, has a ton of carbon relative to a relatively small amount of nitrogen. And so if we were to kill, who are we going to kill off? Who do we want to kill off in class? Let's be equal opportunity. We'll kill off Mackenzie. Mackenzie, are you okay with that?
Sure.
Okay, so we kill Mackenzie and we lay her out in a field. And at the same time we do that, we chop down a tree and lay it out in the field. And the tree trunk that we put in the field weighs exactly the same amount that Mackenzie does. If we come back in a year, what will be the condition of that log and what will be the condition of Mackenzie? Mackenzie, what will you be like in a year?
Pretty much bones.
You'll just be all bones. All of your soft tissue will have been decomposed. What will be the condition of the log that is right next to you?
I mean, most of the bottom would mostly be like soft, it'd be partially decomposed.
It might be partially decomposed, but it's going to pretty much look like a log after just a year. And the reason for this is that the decomposers that are going to act on Mackenzie's body have a lot of access to the nitrogen that's there because that nitrogen isn't surrounded by a lot of carbon. And they go to work on that and they do the process of decomposition on things with a low carbon to nitrogen ratio. These things decompose fast. So in a year, you would be gone, you would be compost. But if you have a high carbon to nitrogen ratio, a lot of carbon relative to a relatively small amount of nitrogen, this is a much slower process. And so if we put a dead body out in the field, the dead body of a pig or a horse or a deer or a snake or anything else would be gone in a year, and a tree log, a tree trunk, would still be largely intact. And that's primarily because of this carbon nitrogen ratio. Mackenzie, we're glad that you're not dead.
Me too.
Good. So that's the process of ammonification, going from dissolved organic nitrogen to ammonium. But we can also add oxygen to these things. So once you have ammonia, we can add oxygen to that to create nitrate. And once again, nitrate is one of these polar molecules that tends to get leached out of soils. And so this is the process by which it goes through. We add oxygens and water, but you don't. So let me also say this. I don't expect you to know these chemical equations. Okay, I'm not going to ask you to recreate these chemical equations. But what I do want you to know is I want you to know the name of the transformation. I want you to know what you begin with, and I want you to know what you end up with at the end. The details in the middle, you don't have to worry about, but you should know what the starting point is, and you should know what the ending point is. So the starting point here is ammonia for nitrification, and nitrate is the end point. And nitrate is hugely useful for organisms because that's one of the easiest things to get converted into something like an amino acid for making proteins. So once again, this is all driven by bacteria, and these are specialized bacteria. Once again, Nitrosomonas and Nitrobacter. Their genus names indicate the functions that they have in ecosystems. And these guys can actually compete for plants for access to ammonium, but they also then create a form of nitrogen that is also useful to plants. This is once again a process that needs the presence of oxygen, so it's called an aerobic process, whereas nitrogen fixation is an anaerobic process. And this is ultimately limited by how much of the reactant that you have at the beginning. How much ammonium do you have? And that limits the rate of reaction. The more ammonium you have, the more nitrate you can produce. So another transformation is assimilation. And assimilation is one of these situations where it's a flux, it's a movement of substances, but it doesn't necessarily cause a difference in form. And so once nitrate and ammonium and amino acids and things like this are in the soil, the plants send down roots. And the purpose of the roots of plants is twofold. One of the reasons that plants have roots is because they need to get water. And so these roots are sent down into the soil to absorb the moisture that is in the soil and bring that to the plant. As you know, plants, as they do photosynthesis, they let loose a lot of soil as their stomata open. Now we're revisiting stuff from the first exam. So whenever your stomata open, carbon dioxide comes in, but water leaves. How do plants replenish that water? They replenish that water by absorbing water through the roots. And then the vascular system of the plant takes the water in the roots and brings it up to the stems and leaves. Well, those roots can absorb water, but they can also absorb anything that is dissolved in that water. So nitrate is dissolved with water, ammonia is dissolved with water, and they'll uptake all of those things through the roots. One of the things that can help with this is the presence of another type of symbiosis. And this type of symbiosis is with a fungus. And these fungi are called mycorrhizal fungi. These are fungi that grow in close association with the roots of plants. And so once again, there is another relationship between a microbe, a fungus, and plant roots. But in this case, this association isn't about fixing nitrogen. It's basically about increasing the surface area that is the root by sending out fungal hyphae, these extensions of the fungal body into the soil, which greatly increases the volume of soil that the plant has available to it. And that's how these fungi help out the plant. So these are some examples of mycorrhizal fungi. Some of these are endomycorrhizae, which basically means the fungus burrows into the plant cells. And so rather than being attached to the outside of the root, they actually send their hyphae, these fungal extensions, grow into the root cells themselves. And you can have, there are different kinds of endomycorrhizae, which you don't really need to worry about. But then there are ectomycorrhizae, and these are mycorrhizae that do not grow into the plant cells. They just surround and create a sheath around the root of the plant. But in both of these cases, what it does is it serves to increase the amount of soil that the plant has access to. So part of this network, this thicker network, this is actually the root system of the plant. But this thinner, more diffuse, almost cloudy looking network that goes through the soil out here, these are all fungal hyphae, extensions of the body of the fungus into the soil. When they're out there, they pick up water and bring that water to the plant root. They also pick up nutrients like phosphorus, which we haven't really talked about phosphorus yet, but another important way in which mycorrhizae benefit plants is not only through movement of water to the plant root, but also movement of phosphorus to the plant root. So once again, in this case, the plant's getting what out of the deal? Out of this mutualism. Mackenzie?
More access to like nitrogen and everything.
Yeah, so they're getting access to water and nutrients. What are the fungi getting out of this? Somebody else. Alyssa. What are the fungi getting from the plant? Is Alyssa there? Winston. Winston, where are you? All right, anybody else? What are the fungi getting?
They're getting like environment that they can flourish in.
Well, I mean, they're soil fungi, so they're kind of already in an environment where they live anyway. What were the
Do
bacteria?
they get?
What was that?
Do they get glucose or like nutrients?
Same thing here. So they are getting glucose, so they're getting an energy source. So in the same way that we had the rhizobium and the root nodules of legumes, they're creating nitrogen that the plant is then absorbing through its roots, and the plant is giving up glucose. In the same way, these fungi surround the root. They bring nutrients and water to the plant root, and what they get in return from that is they get a source of energy that they wouldn't have access to otherwise. So here's a basic graphic basically showing here's the root, here's the soil, and what facilitates the plant getting more access to soil is the presence of, in this case, these rhizospheric microorganisms, which includes, in this case, the mycorrhizal fungi. And so down here at the bottom are the mycorrhizal fungi, and some of them grow into the root itself. Some of them are just on the surface of the root. But in all of these cases, the mycorrhizal fungi extend way further out into the soil then the root hairs of the root will extend. So this is the limit of the root hairs. This is the mycorrhizal fungi going out beyond where the root hairs can extend to. And as a result, the amount of soil that this plant now has access to is much greater than it would if those mycorrhizal fungi were not there. All right. Yeah, we still have quite a lot to go through. So let's take a break here until Wednesday. On Wednesday, we'll actually finish up the nitrogen cycle. But you'll see if you scroll through this, the nitrogen cycle gets really complicated. So I want to give us adequate amount of time to kind of go through and pick this thing apart. And then after we're done with that, you're going to have a homework assignment, which is to basically draw a model of how you think Polyface Farms operates from the standpoint of nitrogen. In order to do that effectively, what you really need to have done by then, if you haven't already done it by then, is, and I had already assigned this to you, but I want to reiterate it. The material that you will need for this is in chapter 11 of Poland's book, this chapter called Practicing Complexity. In that chapter, they talk a lot about how Joel Salatin moves his farm animals around his farm. And a lot of that has to do actually with nutrient cycling. And so having read Practicing Complexity will be useful to you in the homework that I'm going to assign you at the end of class on Wednesday. Questions? All right. Let me know if you have any questions about the upcoming exam, and I will see you guys on Wednesday.